Hi, I'm Judy Fitzpatrick, and I'm going to be introducing you to the subject of building microbial communities. One of the questions we get is, why doesn't my new organic plot have more microbes than my conventional crop? And that's what this subject is going to be about today. It's about all the things you have to do to make your the plot you want to be organic, overcome the damages that's been done to your conventional plot. We'll start with the food web. On the right here, you see the conventional food web, and it shows you all the different levels. At the basis is the microbes and the fungi. And then the organisms like amoeba, etc., which feed on them, and then the nematodes, which feed on them, and the microbes, etc. What this pyramid doesn't tell you is that this layer is 1/1000th the biovolume of the bottom layer. And this layer is 1/1000th of that. And this layer is 1/1000th of that. That's the predator to prey ratio, because these feed on these. So almost any time when you look at the literature, what you see, if you have plenty of these, you have plenty of these. If you have plenty of these, you have plenty of these. The real ratios in the microbial food web should look like this, okay? Because all of these above here or something like 1% of what's the microbial biomass that's in your soil. So you see that the microbial biomass is basically the critical base of the food soil web. The plant and the microbes are in a marriage. The plant delivers 30 to 50% of the food it makes, its photosynthate, to the microbes in the root zone of the rhizosphere. From there, the wealth spreads out into what I call the suburbs. Okay. This is a very sophisticated marketplace that has enchanted economics people in terms of how like it is any economic system. On the right-hand side here, you have the brown representing dirt. You have the yellow being very large, much larger than it would be, representing an AMF fungi, which is one of the microbes which can provide nutrients to the plant. What this doesn't show you is that these microbes, that the AMF microbe is often connected to other trees and things. So it will also, it can often trade with many of the plants in the same region. So the hypha of the AMF fungi is out in the soil. It brings in potassium, it brings in phosphorus, it brings in nitrogen, it brings in sulfur and other microorganisms. It can actually make phosphorus that you don't physically measure by chemical tests available, because it dissolves it from rocks. Now, the AMF, unlike some other fungi, inserts itself into the cell. It stays intact, okay? It's not part of the cell, but it lives inside the cell. And its name, Arbuscular, came from some kind of corruption of house, since it forms a little house in there. And it gives out the phosphorus and the potassium and the nitrogen and sulfur, okay? And in return, the plant gives it sugars and lipids and amino acids, okay? The fungus has the ability to hold back on the phosphorus and other nutrients if the plant isn't giving it the sugars and lipids and et cetera that it needs. And in fact, if it's connected to another plant that's willing to give it more, it sends all its nutrients over to the plant that's offering more food. These are very elegant experiments that have really illuminated to us how this whole system works. Another example of how the microbes provide nutrients to the plants in this marriage is rhizophagy. Rhizophagy means rhizophagy, eat, so it's root eating. The tip of the root does not actually have a mouth, but it has openings, and it attracts and brings in microbes, and it attracts them by secreting foods that they like. And the microbe goes in, and as it travels inside the tip of the root, it multiplies and it releases nitrogen and other nutrients, and it actually supplies 40% of the nitrogen that the plant needs. And then the plant goes to lake rootlets, and they stimulate the formation of these rootlets, and they exit and go back into the rhizosphere. Okay? Fill up again and come back and continue to cycle through. And while they're doing this, they're doing other things. While they're in the plant like this, they're stimulating the plant immune system, which is a talk that I've given in other days. This is important because the immune system of the plant does not develop without these microbes. And when we talk about what minerals do, when you give minerals, the plant is not stimulated to take up rhizophagy or to establish cooperative agreements with fungi. It thinks it has what its stimulus is, the need for nitrogen and probably phosphorus, and without those stimuli, it doesn't need the microbes. So we have a divorce. And in conventional farming, the chemicals and pesticides replace the microbes. The microbes that provided the plant with nutrients and its very important immune function. When you supply the plant with huge amounts of mineral nutrients, okay, you are stunting the growth of healthy roots. Because as I said, the plant is sending out signals to the microbes, food, to come in, and when the microbes get in there, they make plant growth promoting hormones which stimulate root growth. They need root growth because they need the food that the root can exude, 30 to 50%. Okay. If you don't have these microbes because you've given phosphate, you don't get adequate root development. And here we see on the control, and this is all stages of roots, okay, top, middle, bottom. And you can see phosphate doesn't inhibit root growth as much as nitrate and ammonia. Look at these pitiful roots. There is no development whatsoever. Even if you at this point you gave an organic stimulant to this plant doesn't have the root area to interact with those microbes. So potassium isn't so bad. So the worst thing is actually nitrates. Now that doesn't mean you can't you can't supply nitrogen to your soil. It means use organic nitrogen or use low amounts of commercial nitrogen such that they can actually be metabolized by the nitrates, by the microbes, and stored in the microbes rather than inhibiting the plant. So it's your job now to repair the results of this broken marriage of the plant and microbe. And the results, okay, the damage to this child is significant. You have poor, perhaps non-existent soil structure because soil structure is created by microbes. And soil structure provides the places that nurture and house microbes. Without the house, they quickly die. So it's hard to grow them without adequate housing or aggregates that make soil structure. You have depleted soil carbon stores. Okay. There's a beautiful correlation between the amount of microbes, the plant growth, and the available soil organic carbon, not the stored humic carbon so much as that what's available. And when that's not available, the plant is required to go to the bank, to the humic matter, the soil organic carbon, and break down some of it so it can maintain itself during times when soluble organic carbon isn't available. A serious problem you have is you have a decimated microbial population. Your population probably should be between 400 and 600 for maximum production in an agricultural situation. You're probably, if you're a mineral farmer, at 50. When you decimate any population, there are certain subpopulations that get hit a lot harder than others. So it's a hard job to build back a population that actually meets the need of a fully developed working population. And fourth, the crops that have been optimized for mineral fertilizers, a lot of them are not the best fit for working with microbes. They've been engineered to work with mineral fertilizers. Some of them have lost the ability to effectively work with the microbes that are there. So you may have to look at your crop too. Now you have to talk about growing the population to create aggregates to make homes. And these aggregates have been destroyed. How is an aggregate formed? It's formed by fungi and roots, and the microbes tend to stick to these things because and they're creating sticky stuff because microbes always want to stick someplace. They do not want to be washed away. And what you can see here, going from the start of the formation, where everything in here is kind of loose, you can see that as the microbes cover this aggregate, the aggregate becomes more aggregated, and that's because the glue of the microbes is holding it together. Now the beauty of this glue is the glue that the microbes make, including the glomalin that fungi make, remains in the soil long after they die. It's part of the soil organic carbon. And so even if when the aggregate, this one aggregate itself breaks up, we still have soil that has structure. And that soil that has structure, we know, can hold up to 50% more water, making your soil much more resistant to erosion and to drought. How do they build soil? I'm just going to show you here a micro photograph of just soil that was in water, and you see big clumps of soil and stuff here, and you have some microbes, little yellow things you'll see here. But most of the microbes are not visible in this picture because they are stuck to the soil. The wonderful thing about these microbes being stuck to the soil is that the nutrient that's in the microbe that's going to be released when the microbe's eaten by the plant or eaten by an amoeba or something is going to remain in the soil. It doesn't wash away and pollute rivers like nitrogen, phosphorus, and potassium do. And when this microbe dies, the microbe and the sticky substance are going to stay around, creating for you soil structure. And you're going to have depleted carbon stores, which means that you've subjected your soil to food insecurity. Microbes make soil organic carbon from plant material. They make it in their body, which can become humic matter, and they break down things, sometimes not all the way incorporating them, and they get stuck to minerals, and they also become part of the humic matter. So we can basically say that soil organic matter is broken down plant material that passes into soil organic carbon through microbes. So it should be obvious to you then that you cannot increase your soil organic carbon or your fertility of your soil without increasing your microbes. And the soil organic carbon has so many different names that it's very, very confusing. But you can think of it as soluble or readily available, or easily soluble and readily available, and stored. And the stored stuff takes energy to get out of, okay? It's necromass, we call it, which is the bodies of dead parts of the bodies of dead microbes. And it's other foodstuffs that were not fully digested that are attached to the soil minerals or particles. And one of the reasons these things, make it into that is that they're harder to digest. So the microbes have to work harder to get the soil organic carbon. Now, over the years, as we have, harvested and taken away the green grass and the grasses and everything that was going back into the soil each year instead of being harvested and plowed away, the microbes in the soil, even when there's a few, they've had to go back into the soil organic carbon, and we have released about 50% of the soil organic carbon into the air as carbon dioxide. So I just want to impress upon you the reliance of microbes on soil carbon resources. It's food security, and sometimes it's called soil organic microbial available. And when you stimulate, like when the plant puts down an exudate and it stimulates the microbes, if there's nothing else for the microbes to go on once they've been stimulated, the cycle doesn't begin. We've reached a dead end, all right. So it's kind of like having one meal and give it to a person, but there's nothing else in the refrigerator for a week. And I love to talk about the microbial population. The microbial population in the soil is remarkably consistent throughout the world, actually. And it's been around for 3 billion or more years. Most organisms have not managed to survive more than 100 million years. These guys are very good at organizing themselves. And time again, we see that treated properly, they regenerate the diversity and all the conditions that are required. So all of this is happening in the plant rhizosphere. And in the plant rhizosphere, this orange part is the root. It has a mucilaginous material around it that keeps the allows the microbes to colonize there, and we see fungi too. And all of these organisms are alive and are feeding from the plant and feeding to the plant. Now, an important issue about this population is it's an extremely interdependent population. Okay. I guess there are guys just go off and live in Alaska and they can survive a year out in the wilderness. A single microbe couldn't do that. Microbes need a lot of friends in order to survive. They're dependent because they have about 1/1000th the DNA that we have. So if we look at that effect of having fewer DNA genes on a cell, okay, and we understand that every cell, human or bacterial, is a gated community. Okay. 50% of the cell membrane is proteins, which control everything that goes in and out and all communication with a cell. They can have fewer communicator cell receptors. They have fewer receptors for what they can eat. So they're very picky eaters, which is why we can't grow most of them. Okay, digestion for a bacteria, just like for us, doesn't occur inside a cell, it's occurring outside the cell. So the bacteria, if it likes, say, if it likes a small protein for food, it has to have an enzyme that will break down that small protein so that it has receptors that can bring in the amino acids. It probably can't, doesn't have enough enzymes to do all of that. So it probably has receptors for the amino acids, but it's relying on the digestive enzymes of other bacteria to prepare its food for it, to make its vitamins for it, etc. So, these cells are very dependent on one another, and you can't just grow any one portion of this population because of their interdependence. So an example of this is when I did this experiment. So I took a regular plate and I took some soil, I diluted it like they do, and I put it on, and a day and so later, nothing. Luckily, there was a big snowstorm and the school was closed for 10 days. And when I came back, I didn't have this many. There were bacteria growing, and they were all different looking colonies, different colors, which means they're different bacteria. And different bacteria kept emerging over 6 months' time. And that's because, the one that came out in the 40th week could only come out when all the others were there and it was getting, nutrition that it required from them. So I think you begin to understand the process that this population has to do in order to grow to a healthy balance. Now we'll talk about the synergy that has to go between your crop, whether it be a cover crop or, a regular crop, between the plant and the microbes. So they're going to talk. Some of these when the arbuscular mycorrhizal provide phosphorus and nitrogen, that stimulates the plant to grow, and it stimulates the plant to, if it wants those things and is growing, to feed the arbuscular mycorrhizal fungi. And if you and you have in here plant growth promoting bacteria, and the plant growth promoting bacteria are going to stimulate the plant to grow. So you're going to get better growth. They're going to stimulate the plant. Why stimulate the plant? They want to stimulate the plant because they want the plant to give them more food. Okay. So as I mentioned before, the heavy use of mineral fertilizers has led to development of crop types that work really well with mineral fertilizers, but they're now cross-backing with older varieties that were excellent at communicating with microbes so that they can get the right amount of synergy between the plant and the microbe. So I just talk about some of the other ways in which the plant and the microbe match. And these are all experiments, which means you can't generalize from just one experiment to anywhere else you go. Okay, so, but this was an experiment when done where people wanted to know what variety or what mix of AMF would best benefit the growth of their plants. And what you see here on the top here is they're looking at the height of the plant. Okay, so this is on the 32nd day, the 39th day, the 46th day, the 53rd day. And this is plant height. And you can see that that this particular group, the clear one, is doing better than all the others, based on the fact that these, like the different dashes and blacks and everything, are different types of arbuscular mycorrhizal fungi that were used. And you can also see that there was the same response for the stem diameter. It's not nearly as dramatic as the other. This research wouldn't tell you what to do. If you wanted to know what was best AMF for your soil, you would have to take a look at it. And you can take a look at it because AMF is very expensive. You can do an experiment in a pot or in a small plot. And at day 14, actually, you can usually easily see the effect of the optimum level of mycorrhiza. And, you know, the mycorrhiza that you're putting down on your soil, they have to get along with the other microbes that are there too, because all of these guys are defending their own territory. So, you know, different strains, different groups, and different mixes will work differently, even like from farm to farm and crop to crop. So if you want to get into it, you know, there is some experimentation to be done. And we check for, you know, if you want to see the mycorrhizal, you got to take the root, shake all the big dirt off, and, you know, comb it to get the mycorrhizal soil there. And, you know, you can see it's loaded, that area is loaded with mycorrhizal spores, and it'll show up on your in your microbiometer test if you're running one of those. And this shows AMF colonization, and it demonstrates to you that it changes as the plant matures. So there were two sites, okay, and they were treated with yellow AMF and a mixture of AMF with plant growth promoting bacteria. And this is the frequency of colonization, how many colonized. And this is showing you day 14. And this is showing you day 42. Now, you might ask yourself, like, why am I seeing this, okay? And remember, this isn't the AMF was bargaining with the plant. I will give you nitrogen and phosphorus, etc., and you will feed me. When the plant gets ready to, it's fruiting, it's harvesting, it's not interested in feeding the microbes anymore. The microbial biomass, as the plant goes, the plant is giving everything it has to its babies. And so you see the AMF go down. They'll sporulate, and those spores will stay in your soil for a year or two. So, if you want a crop, you're going to be planting a crop that benefits from AMF, then you probably would like to use a cover crop that supplies some AMF so that there's plenty, your corn gets off to a good start with an AMF that's adapted to your soil. It's amazing how differently crops interact with soil. And a lot of this just depends on the soil you're starting with, okay? And what's been done to it in the past, et cetera, et cetera. What that means for you is you need to experiment. With NPK, they just give you a formula. Regenerative can't give you a formula. Any biological and any crop you use is going to have to pair with your soil, and it needs to actually pair with the crop that's going to come after it. And this was an experiment done by Carbon Sponge in New York. We work with, they're illustrating to people, how we can store carbon in the soil. And it was a lot, and it was reading around 80 or 90. And we put on, we did this in pots. We put on wheatgrass. We put on a mix of wheatgrass, clover, and rye. We just did clover, crimson clover. We did hairy vetch, and we did winter rye. And as you can see, we got a 600% increase with crimson clover. This is telling you that because of what the soil was able to give it, the best plant able to thrive in this soil was crimson clover, which we know has rhizobia, so probably the soil needed nitrogen. This does not mean that crimson clover would be best for you. We did a study with University of Tennessee, and throughout what they saw was that hairy vetch was the best cover crop overall. Many people find that the best cover crop is a mix of different things. You can check this ahead of time before you go around, doing your whole farm at the same time. And So researchers have reported that by combining some of the characteristics of older varieties with newer, the plant microbe synergism and plant productivity can be improved. So one of the messages we want to take to you is, if you're switching to at a later time to maximize the benefits, you may have to consider changes in the variety you're using. I guess some people ask, why do you want an optimal microbial biomass? And there probably is an optimal microbial biomass for your soil. Well, the reasons are this. It takes an optimal population, or optimal numbers of people within certain subpopulations to perform many microbial functions, such as controlling disease and stimulating plant growth. And a lot of that is carried on by a system called quorum sensing, which I'll discuss next. Microbial biomass testing also will give you more information than chemical testing, because microbes can make nutrients like NPK available, and this does not show up in the chemical tests. Another thing the microbial level tells you is that your nutrients are at the ratio that is optimal for your plant, because the microbes have the same needs for these nutrients as do the plant. And another thing about an optimal microbial biomass is, time and time again, the literature reports and what we see is that microbial diversity increases almost in a straight line as microbial biomass increases. This resilient population has an intelligence that is hard to understand and is able to grow an optimal population if given the chance. Now, I love to talk about quorum sensing, and the best cartoon I could find is one that talks about how you get strep throat, because that is a function of quorum sensing. So quorum sensing can turn on virulence factors, it can turn on growth factors, it can turn on antibiotic factors. So it's a way of the microbes communicating with one another as to what they think they should be doing at a particular time. So down here, okay, we have where my laser is inside the cell. And where these little bacteria are is outside the cell in your throat. You always have streptococci in your throat. Now they begin to multiply. Something in the atmosphere changed, or suddenly you were exposed and you got a huge bunch of them, okay? And they said, ooh, there's a lot of us, we could have fun. So then they turn off some of their genes, because they can't use all their genes all the time, and they turn on their virulence factor, which allows them to penetrate cell walls and go in and access the rich food that's inside the cell. So I wish we should really talk about all the different ways in which quorum sensing is so important for for your soil health. And microbial biomass, I said, reflects what the plant needs. Well, here you go. Here's soil microbial biomass on the y-axis here. And on the x-axis, I have soil carbon content. Look at the correlation. Great. It's 90%. Here's fungal biomass, and you go almost the same correlation. And what you can see is as one is increasing, the other is increasing. These different colors are because are all different habitats. So they did forests, they did deserts, they did grasslands, farms, etc. So each different color is a different one. And I took Emerson and Domsch's, one of their studies, and I just showed the correlation between microbial biomass, it's their data, and the nitrogen levels they measured. And you can see it's a 90% correlation. So, you need nitrogen in order to be able to grow an adequate population. And if you don't have an adequate population, one of the reasons can be that you don't have enough nitrogen. A lot of people are interested in the correlation of pH, okay? And to some extent that's been, oversimplified. So you can see there is a general correlation between lower microbial biomass at the lower pH and higher at the higher, but there's low liars here. And these are probably, microbes that were low in nitrogen, or there was low carbon or something like that. So pH isn't the only determiner of how you're doing. And a lot of times, we just keep hearing low pH favors fungi. And, to a large extent, it's true that fungi survive very well at lower pHs, okay? But here's bacterial, it's done by PLFA, and you can see it's pretty low, below 5, and then it sort of plateaus between 5 and going towards neutral. So it's slightly affected in these areas, and some crops are very sensitive to the pH of your soil. So, we're not going to knock that. But here's another PLFA showing fungi, and basically this one actually shows that fungi also, function at high and low and mid pHs. So I took that data, the pH data that you saw, and the nitrogen data, and from Anderson and Domsch, and I just corrected it for pH. And you can see here that as I go straight across here, the correlation with organic carbon and nitrogen is amazing. Okay. So these are all stable. These were all samples taken from throughout Europe, and they were areas that had been treated the same for like 10 years. So sometimes people, ask us, what is the optimal MBC level for my soil? And the question is, of course, what limits the microbial biomass in your soil? Well, the nutrient level that you have, okay, both the input and the humic matter and the carbon that you can add, okay, are critical. And then your soil composition is going to limit your storage of soil organic carbon. Sand does not hold as much as clay. So soils that have some clay are good banks for soil organic matter. As you saw, your pH is going to make it hard for your microbes to thrive. Compaction is a problem. And compaction is a problem because there's no oxygen, okay? And actually diffusion is slow when things are compacted that way too. Disrupting like plowing and stuff limits the microbial biomass to 2 degrees depending on the amount of disruption. But I just think of it this way, a big disruption in any society, limits the population. Temperature, we'll talk about that. if high levels of salt will level lowers arsenic, other chemicals that are toxins will keep them down. Some crops, are able to be in your soil and really enrich it with microbes. They work very well together. Their synergism's great. And when you have an optimal population, you have created microbial diversity. That means you build up your fungal, meaning that you're creating carbon in your soil, you're increasing your fertility. Like above 0.5, you're doing a really good job. And it takes a while to build microbial diversity. So, you want to keep microbial diversity, which means that you have a functioning society. I just have a few slides here. This is showing you the effect of compaction. And in this one, we're looking at the total bacterial counts, okay? And this is non-compacted is white, compacted is black. And it's very easy to see that you have more microbial biomass in these 6 different soils when they're not compacted. And on this side, they show you the fungal biomass, how it's affected by compaction. And in this particular case, we're not seeing that the fungal biomass is as decreased as the bacterial biomass in compacted soil. And this is just one graph showing some bacteria. So I wouldn't say all bacteria are going to act this way or anything, but just to show you. Here's a concentration of microbes, okay, how many there are. And this is showing you here that as the temperature increases, the microbial biomass is going down. Okay. And it shows you that as the temperature increases, the respiration, or the amount of carbon dioxide that's being released from the soil, is going up. We prefer to keep the carbon in the soil and not release it unnecessarily by respiration. I mean, they have to breathe, but you don't want them breathing so hard that everything you put down is just going back into the atmosphere. Respiration, the Solvita test or whatever, tells you activity. If a cell is under stress, it works a lot harder, it makes a lot more. So we call this the metabolic quotient. And then we divide respiration by microbial biomass. And when we divide it, okay, we get a number. If the number is higher, meaning you have more respiration for microbial biomass, then you're putting more carbon into the air than you're storing in the soil. So what we see is building carbon in the soil is inversely related to how much respiration you have per MVC. So using respiration alone to figure out if you are sequestering carbon is not a good idea. And here again, I'm going to show you how microbial biomass varies over the season. Now, different methods will show you different ways. So if you're trying to measure something, you want to stick with the same measure. Now this shows you this is microbial biomass. This is not okay from the literature. Down here who did it. And in the spring, this is the microbial biomass, and this is the respiration. So you can see that the respiration, isn't always the same. It's often not the same as the microbial biomass. In this case, if you take the CS experiment, respiration was really high for a low number of microbes. In the summer, we see the same thing here for those first three respirations really high and microbial level is low. Microbial level is lower in summer. Remember we said heat lowers the microbial, increases respiration. Now we get into autumn, and this is interesting because you see a large increase in microbial biomass in autumn. And this is no doubt due to the fact that in autumn, you've just harvested, you've left litter, and there's roots. So the fungi are really chomping down, multiplying, breaking down all this stuff. And fungi are very high in carbon, high carbon to nitrogen ratio, and there's a much lower carbon to nitrogen ratio for bacteria. So now we have a fungally dominant scenario, and because fungi are very efficient at using carbon, what we see is the respiration is low. Okay, so it's the lowest respiration we have, even though we have lots of fungi there. So to summarize in a way, your organic or sustainable system is entirely or largely dependent on the microbial community for immunity to pathogens. A big study of hundreds of farms in Europe over like 10 years showed that organic farmers used 97% less of any type of pesticide. A wonderful up part of this is that the organic vegetables that are not treated with pesticides, that fight off disease by themselves, they generate antioxidants, and the antioxidants they generate are what gives food its higher nutrition and its excellent taste. Okay, the plant is dependent on them for its minerals and nitrogen, for digestion of litter to increase soil organic matter. The microbes feed the plant information about soil conditions that allow the plant to adapt. An example is AMF. When you have a drought, AMF signals the plant to bring a bunch of sugars down into its root. The root actually swells up if it has AMF on it, which increases the osmotic pressure inside the plant, and it can suck extra water out of the soil. So it's wonderful for making these plants more drought resistant. You're dependent on you need to create soil structure and increase the water holding capacity to double, which can't be emphasized enough. This is a great advantage. If you're not watering, it makes you more drought resistant. If you're watering, you can save on water. You get protection from erosion by increasing soil structure and also by covering your soil with a cover crop instead of leaving it bare, where rain can wash it away. And you're sequestering carbon in the dead and alive microbial biomass. So you're saving carbon, a lot of carbon. If there's one elephant, they say, of microbial biomass in every acre of soil, then if you double it, you got 2 elephants of carbon. That's a lot that you put in. You don't have to wait 100 years for that. So the key to transitioning is starting by building that microbial community. And we can't give you a formula for that, like how much NPK. It's going to depend on matching your soil, your climate, and your crop. And the microbial community can do this if it's given the right foods and conditions. And you can tell very rapidly, like within a week, if you gave it the right foods by measuring your microbial biomass with the microbiometer. And you can use optimal fertilizers and amendments, and you can tell if they're working, if this is the right one for you, by measuring microbial biomass. And before we end, I'd like to just put in a plug for cover crops. A cover crop is the best way to get started in regenerating soil. The cover crop can double, triple, quadruple the amount of microbial carbon you have in your soil. That's a significant amount of carbon. And it can create lots of structure, so you need less water. And in fact, it's a fertilizer. The roots and the grasses that were there are going to stay, and they're going to cover your soil during the winter and the rainy springtime when you can lose soil from erosion. And then not having to put down as much fertilizers, especially if you even have to use a little of the NPK, you can use it much more sparingly, and you can prevent pollution. So this is why we believe cover cropping is the best way to start on a regenerative pathway. And I'd like to thank you very much for participating in this webinar. If you'd like more information, please contact us at info@microbiometer.com. Thank you.